In this video, we will be going through the preliminary steps for running a qPCR. It is important that all preparatory work is done in a sterile environment, either in a BSC or over a Bunsen burner, as qPCR is a sensitive technique that can amplify minimal contaminations. First, ensure that reagents, mixtures, and samples are kept on ice throughout the duration of the qPCR setup. This may include your sample, reverse and forward primers, and your fluorescent dye. A master mix can be prepared for every sample that requires the same primer set. Your master mix will contain a fluorescent dye such as cyber green, forward primer, reverse primer, distilled water, and your external control. After adding these components to your tube, make sure to pipette up and down vigorously to ensure that all components have been mixed well. These master mixes can then be aliquoted to their designated well of the qPCR plate prior to adding the template or sample to the well. Ensure that an additional amount of master mix is made in each tube to account for errors that may reduce the overall volume. If you're planning to run multiple samples simultaneously, it may be helpful to prepare a plate template prior to pipetting, allowing you to keep track of which sample goes in each well. Make sure to pipette the master mix and sample to the very bottom of the corresponding well, being cautious not to pipette sample onto the walls. If equipment is available, use a plate spinner prior to running the qPCR. When you're mixing, make sure to use smooth motions to avoid splashing of any sample out of the well or generation of aerosols that may cross-contaminate the other wells. Keep all reagents, mixtures, and samples on ice throughout the remainder of the experiment to prevent potential degradation of reaction components. After the master mix and samples have been pipetted into the wells, you will be ready to seal the plate. Supplies needed include the qPCR plate seal and film sealing roller. Peel the wax paper layer away from the clear film and place the edge of the film along one edge of the PCR plate, in a similar fashion to putting a band-aid on. Continue peeling away the wax paper as you apply the film over the wells. Take your rolling device and smooth out the film over the plate, using firm pressure while rolling. Proper sealing of the plate will minimize evaporation and allow for plates to be stored after the run is complete. To begin setting up the qPCR program, open up the laptop located in the designated lab room. Turn the computer on and start it up. To turn the thermocycler on, reach behind the back of the machine and flick the switch. The machine will take about a minute to warm up. In the meantime, you can load the qPCR program on the computer. The Bio-Rad CFX Manager software can be found as an icon on the desktop. Double-click to open it. Once the program starts up, you will want to click on the User-Defined Protocol button. In the top left corner, you can then click the Create New button to customize your protocol. A screen will pop up showing the steps of the run. As with regular PCR, the length of time and temperature for each step can be programmed according to your needs. The parameters for each step can be changed by simply highlighting the preset value and typing in a new number. It is important to double check that there is a plate read step in place after each cycle. This step instructs the machine to record levels of fluorescence in each well after every round of cycle and is important in determining CQ values. A typical number of cycles for qPCR is 40 cycles. To check the purity of your qPCR products, it is recommended to perform a melt curve at the end of your qPCR run. To insert this additional step, click on Step 4 and then press Insert Melt Curve button on the left panel. A new step indicating the melt curve temperature should appear. These temperatures are also customizable. Once you are satisfied with your parameters, you can click OK in the bottom right corner and save your program for future use. Once saved, clicking Next will move you into another screen showing a plate template. At this point, you may label each well with your given sample. Navigate to the Express Load drop-down menu. Pick an option that lists 96 well plate. Depending on which fluorescent dye you are using, you may need to use a different setting. If you are using cyber dye, click the Quick Plate 96 wells cyber only drop-down option. Click Next in the bottom right corner again to move to the next screen. At this point, all your settings are in place and you're ready to put your plate into the machine and start the run. Click Open Lid to lift the lid of the thermocycler. Once the machine opens, you can slot the plate into the appropriate mold, being careful to keep the plate level and minimize any sudden movements while doing so. Once the plate is securely in place, you can press the gray button on the front of the machine or click the Close Lid button on the computer to reseal the instrument. Click Start Run. The program will prompt you to pick a folder or location to which you would like to save the run data. Label your run appropriately and then click Next. The program will open a visual run tracker for you, where you can see which step the run is at any given moment in time. For a few minutes after starting the run, the panel near the bottom will say that the run is initializing. Eventually, that panel will change to indicate that the machine is running and that your time steps have started. At this point, you can change your view if you wish to view the fluorescence levels in real time. To do so, click on the real-time status at the top of the window. This will open up a screen that will graph the fluorescence over time. In the early run stages, this will present as a straight horizontal green line, indicating background fluorescence. When the run is in progress, the light on the front of the thermal cycler will glow green, indicating that it is in use.